So we've been taking a look at how a cell makes proteins. And again, basically the information on how to make a protein is found in the cell's DNA. Making a protein, though, involves two steps. The first step involves reading the information on DNA in order to construct a messenger RNA molecule, and that process is called transcription. And the second step of making a protein involves using the information on messenger RNA in order to then string together amino acids to make a protein, and that process is called translation. We've showed you what all is required in order to do transcription, and we also showed you what all is needed in order to do translation. It turns out, though, that after a cell has completed stringing together a bunch of amino acids to make a protein, things have to happen then to this string of amino acids before that protein can be a fully functioning protein. And so what we'll show you next is what all does a cell do to this protein after it's been made in order to make it a fully functioning protein. And so to differentiate between just a string of amino acids that's not functional yet versus a fully functional protein, typically what a protein is called right after translation is finished. It's called an immature protein or a polypeptide. Again, immature meaning that it's just the amino acids that make it have just been strung together, and the protein hasn't been modified yet so that it's not fully functioning. And again, what we're going to do is show you what all happens to, or what all a cell does with a string of amino acids in order to make it not an immature protein, but a fully functioning mature protein. And so here we'll show you three common things that a cell ends up doing to its polypeptide in order to make it a fully functioning protein. So in this picture, this shows a ribosome and then a messenger RNA, and then here's the string of amino acids that it's making based off of the information on that messenger RNA. One of the things that a cell does with the string of amino acids in order to make it a functioning protein is that it adds carbohydrates to it in a process called glycosylation. So generally whenever you see this prefix glyco, that refers to sugar. So this term glycosylation means that a sugar was added to something. And so in this particular case, a sugar was added to a polypeptide in order to make it a more fully functional protein. So in words, glycosylation just means adding carbohydrate groups to something. In this case, we're adding carbohydrate groups to a protein. Other things that could happen to a protein, a polypeptide, in order to make it a fully functioning protein is that actually sometimes a cell ends up cutting up this string of amino acids. And whenever this occurs, whenever a protein is cut up, that process is called proteolysis. So if you remember, lysis means to cut up, proteo means protein, so literally proteolysis means cutting apart a protein or a string of amino acids. And then one other common thing that cells do to a polypeptide in order to make it a fully functioning protein is that they add phosphate groups to it. And so we've already mentioned what this term is called. It's called phosphorylation. So again, that just means adding phosphate groups to something. So you might be wondering, well, how do these activities make a polypeptide a fully functioning protein? Well, basically, glycosylation, what this does, by adding sugar groups, carbohydrate groups to a protein, those carbohydrate groups basically act as identification tags. So for example, if this is a protein that's going to be secreted by a cell, by having these carbohydrate groups on there, it kind of lets other cells in the body know what this protein is going to do. It lets other cells in the body identify this protein as something that's made by our own body instead of, for example, by bacteria or viruses, something that might be potentially harmful. If you're considering these two things, so this seems kind of counterintuitive. So we're trying to make a fully functioning protein. It seems that cutting it up would make it non-functional. Well, it turns out that if the protein is cut in certain correct spots, it actually makes it so that it helps the protein fold in the correct way. So cutting the protein basically helps the protein fold in the correct way. And the same thing with phosphorylation. So adding phosphate groups places extra negative charges on a protein. And we saw earlier in the quarter that by adding charges to a protein, that could potentially help it fold in a particular way. So when you're considering proteolysis and phosphorylation, basically the reason for doing these things is that it helps the protein fold properly. And again, if the protein is folded properly, then it's more considered a fully functioning protein at that point. So back to the big picture here. So again, we're talking about how proteins are made. What we just said is that typically cells do stuff to this string of amino acids in order to make it a fully functioning protein. For example, the cell does stuff to this string of amino acids in order to make it fold properly. And so now after that string of amino acids has become folded in the correct way, now it can be considered a functioning protein or a mature protein. It turns out that proteins do lots of different things for a cell. So you might remember that some proteins act as enzymes, some of them are secreted from a cell and act as messenger molecules, some act as antibodies to protect us against bacteria and viruses. Some proteins act as pores that let things through the membrane of the cell. So proteins can do lots of different things, which means that proteins are found in lots of different places, right? Enzymes are found in different spots in a cell. Pores are found in different spots of the cell. Antibodies are actually secreted by a cell. So in order for this mature functioning protein now to be able to help out the cell, to be able to carry out its function, it has to be delivered to the right spot. So that's what happens next. It has to be delivered to the appropriate location, either within the cell or it's secreted from the cell if that's what its function is. And so what we'll do next is show you what all is involved in helping to deliver a protein to its appropriate location within the organism. We'll mention right off the bat that the thing that plays a big role in helping to deliver the protein to where it belongs is something called a signal sequence. And like it says here, a signal sequence, it's basically an amino acid sequence that's found on the protein itself that kind of tags it, that lets the cell know where to deliver the protein so that it can do what it was meant to do. So what we'll do is that we'll show you some examples of different places where proteins can end up and how they end up going there. So here's a picture of a eukaryotic cell. So here are different organelles that you find inside of a eukaryotic cell. Whenever a protein is made, the thing that directly produces the protein, the thing that has the information to directly produce the protein, is a messenger RNA molecule. And so like we saw not too long ago, in order to use this information to make a protein, there have to be ribosomes involved. And so it's at the ribosome then that information on the messenger RNA is used to string together a bunch of amino acids to make a protein. It turns out that there are some proteins that don't have a signal sequence on them. So for example, this one does not have a signal sequence on it. And so whenever that's the case, if a cell ends up making a protein that has no signal sequence on it, here's what happens to it. When it ends up folding, again, if it has no signal sequence on it, what that does is that it causes the protein to stay in the cytoplasm. Right? So there are some proteins that maybe act as enzymes that do something in the cytoplasm of the cell. Maybe it's a structural protein that will help maintain the shape of the cell, and those are found in the cytoplasm. So there are several different types of proteins that do their job just within the cytoplasm of the cell. The way that the cell knows to keep it within the cytoplasm is that these are proteins that don't have a signal sequence on them. Right? So what we're saying here, the bottom line is that if a protein has no signal sequence on it, then the cell knows to make it stay put in the cytoplasm. That's where it's going to do its work.